Hello friends, welcome to the channel. In this video, we will study about the microbiology of wine. Wine is an alcoholic beverage made from fermented grapes. Grapes ferment without the addition of sugars, acids, enzymes, water or other nutrients, as yeasts consume the sugar in grapes and converts it to ethanol and carbon dioxide. The science of dealing with the wine is called enology. Now, let us look into the history of wine. Wine has been produced for thousands of years. The earliest known evidence of wine comes from Georgia, where 8,000 years old jars were found. Traces of wine have also been found in Iran with 7,000-year-old wine jars. Wine was also consumed and celebrated in ancient Greece. Wine is basically of two types on the basis of its color, that is red wine and white wine. Red wine is a type of wine made from dark colored or black grape varieties. The actual color of the wine can range from intense violet, typical of young wines, to brick red for mature wines and brown for older red wines. The juice from most purplish grapes is greenish white. The red color comes from anthocyanin pigments present in the skin of the grapes. Next comes the white wine. White wine refers to the wine whose color can be straw yellow, yellow green or yellow gold. It is produced by the alcoholic fermentation of the non-colored pulp of grapes, which may have a white or a black skin. It is treated so as to maintain a yellow transparent color in the final product. The top fermenting yeast, Saccharomyces cerevisiae, is used for the fermentation of grapes in the production of wine. In the process of production of wine, yeasts are grown in the winery by inoculating about 1 to 5 liters of fruit pulp, that is known as must, from the laboratory culture slant, and then serially transferring the inoculum to successively larger volumes of fresh must until a quantity suitable for bulk fermentation is obtained. The grape juice is treated with bisulfite to kill undesirable yeast and bacteria. Fermentation is done in vats which are usually of wood or stone and vary in capacity. Vat is covered to maintain anaerobic conditions that encourage fermentation and discourage growth of acetic acid bacteria, which could convert the wine into vinegar. A favorable temperature for fermentation is in the range of about 10 degrees centigrade. A low acid content favors a good effect on the flavor, color and stability of the wine. After the partial fermentation, the racking operation begins and continues throughout the remainder of the fermentation period. In racking, wine is siphoned off from the sediment, which consists of dead yeast cells and other materials, which settle at the bottom of the vat and is transferred to a fresh vat. This procedure is repeated several times until the desired clarity of the wine is obtained. Fermentation continues until the alcohol content is about 12%. The wine is then stored in completely filled and sealed tanks for aging. Additional blending fortification with additional alcohol may be required before the wine is bottled.